Welcome to the webinar on ImageLab software. Thank you for taking time out of your day to join us. First, let's look at the topics we'll be covering. Today's webinar is ImageLab Learn Total Protein Normalization. We'll focus on the process of analyzing data from chemiluminescent blots using ImageLab software. We'll be comparing total protein normalization to housekeeping protein normalization. We'd like to provide you with simple steps to successfully and accurately analyze Western blot data. ImageLab makes this data analysis easy because it's automated wherever possible. Let me also introduce myself. My name is Kate Clark. I've been a member of the Bio-Rad technical support team for over 12 years. I'm excited to share some tips for using total protein normalization to more accurately quantitate chemiluminescent data. So let's begin. Let's begin with the challenge. How to get accurate band quantity from chemiluminescent blots. The Western blotting process can introduce artifacts into data. For accuracy, it's important to minimize these artifacts. First, we'll look at the challenge in three broad categories: getting good blot quality, getting good image quality, and getting good data quality. We'll then review solutions to these challenges, including TGX stain-free gels and protein normalization in ImageLab software. We'll finish by using ImageLab software to normalize data with total protein normalization, and we'll compare it to housekeeping protein normalization. Now let's get started. I want to start by taking a step back to make sure that we're all on the same page about how imaging software measures band quantity. In ImageLab, band quantity is measured by finding the volume of each band. As you know, volume is a three-dimensional quantity. In ImageLab software, volume is the sum of intensity levels within a defined area. For example, on the left is a 1D view of a chemiluminescent blot image with 3 lanes, and one lane, the band of interest is boxed in green. On the right is a 3D view of that same band with the intensity levels is shown as the height of this mountain peak, and the intensity levels go up with this red arrow and they vary. And the area of the band is shown with the green line around the area. Therefore, the volume of this band is the sum of all intensity levels within that band area. Let's look at a blot image to see what we're looking for now. We're looking for changes in volume in our bands of interest from lane to lane. The challenge is to make sure the differences in band volume we see in the final data are real, meaning that the differences in volume from band to band are not due to artifacts such as uneven loading or saturated signal. Again, on the left is a 1D image of a blot stained for total protein. On the right is the 3D view of the same blot with 2 numbered lanes. And notice that the peaks in red have the opposite trend as the peaks shown in black. This suggests there could be differences in expression between the 2 lanes. What we need to know is if these differences in detected signal are due to actual differences in expression levels of that protein. This is an important variation in the data. On the other hand, if these differences are due to an artifact, we need to prevent it or to normalize it. Artifacts in signal can occur throughout chemiluminescent blotting process. The biggest challenge is to minimize and to remove them whenever possible. Let's look at the sources of these artifacts, these challenges. The chemiluminescent blotting process has these 3 broad challenges. The first challenge is to get good, consistent blot quality. This involves making sure there's consistent gel loading, consistent gel transfer to the blot, and consistent antibody signal. The next challenge is to get good image quality by making sure there are consistent loading controls for the sample and minimal antibody and image saturation. The final challenge is to get good data quality. This involves having good tools to normalize the data with a loading control. So let's look at loading control options now in more depth. It is common to use a housekeeping protein as a loading control. I did this in my own thesis research. Here are 3 challenges for this type of loading control. First, it's critical to verify that the housekeeping protein is consistently expressed from tissue to tissue and from treatment to treatment. Second, to avoid signal saturation with housekeeping proteins, it's common to image the band of interest and then strip and reprobe the blot for the housekeeping protein. This requires extra time, and the stripping and reprobing process can create an uneven blot signal. Finally, to avoid that unevenness and saturation, as well as avoiding stripping and reprobing, it's possible to balance all the loading concentrations of all the proteins in one blot. And this would take extra time as well to set that up. In contrast, let's look at the challenges with using total protein as our loading control. Total protein loading controls are gaining popularity and provide an alternative to using a single housekeeping protein as a loading control. Total protein stains are often easier to use and more reliable for normalizing blots. We've recently found over 200 studies where the total protein expression was more consistent when compared with any single protein expression level throughout an experiment. So what are the challenges? The first is to find a total protein stain within the relevant linear range that is typical for blotting. Typical blot stains that are linear within the right range are Kamasi, Amido Black, and Cipro Ruby. Second, when using a total protein stain, requires that it be compatible with the chemiluminescent blotting process. For example, Kamasi is not compatible with subsequent chemiluminescent blotting on the same blot, and so it requires that you run 2 parallel blots for each experiment, which isn't ideal. Finally, staining and destaining needed for many total proteins can cause uneven staining in the blot image and take more time. For example, both Amido Black and Cipro Ruby blot stain, which have a good signal, but they require destaining before imaging. And in my thesis research, I found that Ponceau in general was rather inconsistent as a total blot stain. For all these challenges, TGX stain-free gels provide a good option. Stain-free gels are fully compatible with the blotting process. The stain is fully compatible also with the common protein loads on blots with a linear range of 10 to 50 micrograms of total protein. And because there's no staining and destaining, there's no risk of uneven staining. So let's look a little bit at how TGX stain-free gels can solve challenges for chemiluminescent blotting. At Bio-Rad, we've supported Western blotting products for decades. We're always looking for ways to shorten the process and to strengthen the data. Several years ago, we introduced the Bio-Rad TGX stain-free gels to solve many Western blotting challenges. These proprietary gels contain a fluorescent tag. The tag is activated by UV light and covalently binds all protein in the gel. The tag then transfers to the blot with the protein, allowing you to view all the protein on the blot. For this webinar, we'll be using 3 images from a stain-free blot. This slide provides an overview of the process we use to collect the images. We call this the V3 Western process to emphasize that you can visualize, verify, and validate your raw data using TGX stain-free gels. So here are the steps. In step 1, we ran the TGX stain-free gel with standard tris glycine buffers. After running the gel, we activated to couple the fluorescent tag and took a gel image, and that's step 1 right here. This image is useful because it allows you to check the gel to see how it ran before going on. In step 2, we transferred the proteins to the blot. We took an image of the blot to verify that the transfer was complete. That's step 2 right here. In this webinar, we'll be using this image as the total protein loading control for normalization. If you set up your transfer for the first time, it's helpful also to take a picture of the gel at this point to make sure that all the protein you wanted was transferred out of the gel and onto the blot. In step 3, we block the blot and we did the washes and the antibody reaction as required by the primary and secondary antibodies. And in step 4, we added the chemiluminescent substrate to identify the bands of interest. We waited 5 minutes and took an image. This image contains the bands of interest. For good chemiluminescent digital images, be sure to use a camera-ready chemiluminescent substrate such as Clarity Western ECL. At this point, you can strip and reprobe the blot if you're using a housekeeping protein for a loading control, or if you need to visualize more antibodies on the same blot for any reason. In the final step, step 5, we'll normalize the data in ImageLab software, and when we open the 2 blot images and use the normalization tool. For total protein normalization, we're going to use the stain-free blot from step 2 and the chemiluminescent blot from step 4. When we use the housekeeping protein normalization, we're going to use 2 chemiluminescent blots from step 4. One last important note. If you expose the glass in your imaging system to any ECL reagents, be sure to wipe it clean with a wet towel and to dry it. When dried on glass, all ECL reagents can etch the glass, leaving a permanent mark that interferes with image quality. So now that we have our blot images, let's review how the normalization works in ImageLab before going into the software. ImageLab software has a normalization process that involves 3 quick steps. First, you create a multi-channel image. In ImageLab, this image links the data from all the images into channels for analysis. The second step is to identify the lanes and bands. In this step, we'll frame the lanes, we'll subtract the background, and we'll detect all the bands we need for analysis. Finally, we'll use the normalization tool to normalize the data. We'll select the total protein, total lane protein, or housekeeping protein channel to calculate the normalized volume. So before we open ImageLab to go through these steps, let's take a brief look at the calculations needed to normalize data. To calculate the normalized volume, ImageLab uses a loading control you select, either total lane protein or housekeeping protein bands. Here are the formula used. The normalized volume itself is the raw volume of the band of interest multiplied by a normalization factor, and that's shown in this top formula. The normalization factor, shown here, is calculated from the loading control. So you'll use a reference volume, and that is divided by the raw volume in each lane. For the reference volume, ImageLab uses the first lane on the blot, as long as it's not a molecular weight standard lane for the loading control. For all normalization factor volumes, the source depends on the type of normalization. For instance, if you're using total lane protein, then ImageLab uses the volume of the lane, and the reference volume is the first lane. So it's the volume of the total first lane. For housekeeping proteins, ImageLab uses the volume of the band, the housekeeping protein band, and the reference volume is the first band in the first lane that's not a molecular weight standard. These formula can take a little bit of time to digest, so it helps to see them in action. So now let's go into our software and see how it works. Today we'll be using these 3 images. Again, they're all from one stain-free blot. The first image is the stain-free down here. That one shows the total protein that was loaded on the blot. And the next 2 are our chemiluminescent images. These 3 images were taken of the same blot again, and there are a total of 9 lanes loaded. The data are taken from rat brain tissue, and each lane represents sequential stages in brain development. When I'm working with new images from a new sample source, I find it really useful to take a quick look at both the 1D and 3D views of the images before doing analysis. This gives some idea of what to expect out of the data and allows me to check for problems. So the first image we'll look at is the chemiluminescent blot of our bands of interest. That's our myelin basic protein here. It's expressed at different sizes at different time periods during development. For the purpose of this webinar, we'll only be looking at the data from this top band, which changes and is in every lane. So in the 1D view, you can see that there's definitely changes in the amount of protein detected. You can visually see that they're lighter on the first lane and the last lane in particular, lane 7, and these middle lanes are very dark. Let's look at the 3D view. We'll blow it up a little bit here so we can see the differences. And again, you can see that there's a fair bit of difference here, and overall the volumes in these middle lanes are quite high, whereas the first and last lane there and there are very low. Okay, the second image here is our stain-free blot I'm going to look at next. This image again shows you the total protein that's in each lane, that was loaded into each lane. And we can see that overall a similar amount of protein was loaded into each lane, although there's a little bit lighter levels here. And this lane is a bit narrower, which means it might have a little less protein as well. If we go to the 3D view on this one, and again I'm going to extend it so that we can really see what's going on here. We can see again that looking at the height of the peaks, lane 7 is low and lane 1 is also low. And if you look at the width of the peaks, you can see that lane 1 is particularly narrow. And so there may be some differences in the total amount of protein that was loaded onto this image. All right. The third image here is another chemiluminescent blot, and this is an antibody to beta-actin. We'll be using this for a loading control for a housekeeping protein normalization. In the 1D view, there is also variation that shows up from lane to lane. In particular, these first 2 lanes are particularly smeared, which suggests there's some overloading of that protein. And the last lane is a slight bit of smear as well, so it could be rather high also, which is a little concerning when you think that this is the normalization lanes, that there's such variation. Let me look at the 3D view now. Let me arrange it back here. And we can see what the smear looks like. It definitely adds volume to this first lane. And you can also see that there's differences in the height of these band in each lane, which again suggests that there's some variation in actin expression from time period to time period in rat development. As it turns out, there is some debate in the literature about using actin as a loading control for rat brain development studies, and that's what prompted the collection of this data. So now we're ready to normalize the data. We'll begin by using the housekeeping protein to normalize the myelin basic protein. And so we'll only need these 2 images. I'm going to close the stain-free image for right now. The first step, again, is to create a multi-channel image. So with these 2 images open, I can select File, Create Multi-Channel Image. I'm going to then load the images that are listed here into the multi-channel image, and I'll double-click the first one, which is my protein of interest. That's my myelin basic protein. It's added to the red channel. And my housekeeping protein or loading control, I'm going to add into the second channel, which is my green channel. You can see in the overlay image here, these 2 images overlaid, that they're slightly out of register. This isn't a problem. It just means that the blot was taken out and then put back in before imaging again, so it's slightly off center from the first image. And there's no problem with this. ImageLab can handle this as long as you use the same lane frame for every image, then it can line up everything correctly. So click OK, and it creates a multi-channel image showing both of the two blot images, my actin and my myelin, and a multi-channel, the overview image. I'm going to close this one because I don't need the overlay for analysis. So close that and get it out of the way. And I'll blow these up a little bit. So step 2 in this process is to identify all the lanes and bands. And for that, we'll open the lane and band tools here. First thing is to frame the lanes. Now there's a couple things to consider when framing lanes. And what you want to do, since we're going to use the same lane frame on both of these images, I need that lane frame to include all of these bands here and all the bands here. So I definitely want it to go up high enough and down low enough to include all of these. So the next thing I want to consider when I do that, so I'm going to choose this one to start with because it has more bands in it, then it makes it a little easier to line everything up correctly. With one band, it's a little harder to create a frame. And so I'm going to use the manual setting to do that. I know that there's 9 lanes in here, so I'll type in 9. And ImageLab creates a lane frame here. And we'll get this smaller down here and resize it to fit. What I want to do is make sure that each band is between these two red lines so that there's no band that's overlapping any of the red lines. I want to make sure that these bands are going to fit within this frame. And I want to make sure, this is the third thing to watch out for lane frames, I want to make sure that the top of the lanes are in some background area, not in a band, and the bottom is the same. I want it within the background of this blot. So if you can see the edges of the blot, that helps. On an image, I can just barely see them so that I know that the bottom here and the top here are within the background of the blot. So now I have the lanes, and I need to go to the background settings. To do this, what I usually do is click one of the lanes and I open up the lane profile. And you can see in the lane profile, you can see where the background is. That's that gray area underneath these peaks. What I want is that the background be very nice and flat along the whole background of the image. So I'm going to shift this rolling disk size, this disk size of 10 is a little bit small for this image. It's rolling sideways through here, and I want this to be very flat so that it's only subtracting background and not any of the noise that's above the background. Typically, for a nice clean blot or a clean gel, I find that a rolling disk size of about 70 is a good way to go, and so I'm going to start with 70 and click Apply. And you can see that the background flattens out there. So the noise above background is considered as well as the peak that I'm interested in. So I can click through using the lane tool up here and make sure that that background setting is working on every single lane. It looks fine. And so that's all set. So now I have the lanes set, framed, and I have the background set. And I can simply copy this frame using Control-C on my keyboard and click over here, and Control-V will paste it onto the other image. Now remember, these are out of register, so I'm going to have to click on the either resize or adjust and click in the middle so that I don't actually resize it, but just shift drag this into register with this image so that, again, you want background below, background above, and all the bands are included in the lane itself. It looks like it needs to be a little more this way, but I don't want to miss that one. So getting these two within the frame. So now we have the same lane frame on both images, and we're ready to go to the next step, which is to set the bands for the framing here. On these two images, I'm going to need the bands on both this image, because this is my normalization bands, and this image, which is my data. So I'm going to use the detect bands. Since I'm on the actin, I'll start with the actin bands. I'll click detect bands. Since these are prominent bands, I'm going to use the low setting. It'll be fine. And I'll close that. So all these bands are set up. If I want to, I can double check that the band profile is good by clicking on the lane profile and looking at how that looks. Right now for time, I'm not going to go through that right now. I'll click over to my other image. And for this image, I just need these top bands, but I'll start by detecting all the bands really quickly. They're all there. And then I'm going to use the delete button here to delete the ones I don't need. There we go. So I have all my bands of interest and my housekeeping proteins. Again, you can also see that you can adjust the bands here if you needed to. So the third and final step is to normalize the data. Let's go over to our normalization tool right here and click normalization. First step is to select the normalized channel. That will be my green channel, which is my actin over here, the green channel. And I'm going to select the method of normalization, that's housekeeping proteins right here. So my data is ready now. I'll click on the analysis table and open up the data. And you can see here, oops, I don't need all of these, so I'm going to move everything out of here. Pen label, molecular weight. Don't need any of that. Don't need any of this. I just need those data. Okay, there we go. So I have the volume of each band, I have the normalization factor for each band, and I have the normalized volume. The first band here is my red channel. That's my band of interest. And the second band is my actin band. And you can see if I click over to the first lane here, you can see the normalization factor is 1 in lane 1 because that's where we normalize everything else too. So let's export this data real quickly to Excel. I'll open this up. I'm going to sort it to get the lanes. So that the first set of bands is my bands of interest. And I'll make this bold and open this up. And you can see that the last set of bands is my actin channel. They're all normalized to each other, to the first lane, basically to itself. And so all the normalized volume, which is this third lane, or third column rather, is the same. So actin starts out with this raw volume multiplied times the normalization factor for each one, and you end up with the same normalized volume that's going to be applied then to your bands of interest. So these normalization factors are then applied to the bands of interest up here. And what we see is that for each raw volume beyond lane 1, where the normalization factor was created, we had to increase the amount of protein in each raw volume to get a normalized volume. And what that means is that the actin, for actin, the first lane is actually higher than all the other lanes because these lanes all had to be increased to get the same normalization factor. So myelin basically, normalizing to actin rather, shifts our myelin volume higher. So that's what happens when you use a housekeeping protein to normalize the data. So now let's use the same myelin image and normalize with a stain-free image for total protein loading control. So I'm going to close this and close that. We'll close our multi-channel image and the data since we don't need that. I'm going to close the actin also, and then we'll open up our stain-free image here. So now we're going to go on to total protein normalization. Go through those steps. So the first step is to make a multi-channel image, and we'll go up to File, Create Multi-Channel Image. We've got our two images open, so they'll both be in there. I'm going to put my myelin basic protein in the first channel again, the red channel. Put the stain-free blot image in the second channel. And you can see again that these are where one image must have been taken out of the imager and then placed back in to get the image. So they're just slightly off register, but again, that doesn't matter as long as we use the same lane frame. So I'll click OK to create the multi-channel image, and we'll close the overlay view since we don't need it. Increase that. Okay. The second step is now to frame the lanes. We'll go to the lane and band tools. Click over here. Now when we're considering which image to use for lanes, in this case I'm going to choose the total protein because in order to normalize I need all of the protein in every single one of these lanes. So I want the lane frame to include everything that's on this blot. So I'll start with framing those lanes, and we're going to use automatic on this since Image Lab can see the entire lane there. Let me resize this quickly. We're going to move the bottom here up so that it's within the background of the blot, and move the lanes over just a little bit to allow them to pick up these really heavily loaded areas on this blot image. There, so we have the top up at the top above the protein, and the bottom down at the bottom below the protein, and all the bands are within the lines of each lane. Well enough. And again, we're going to go in and set the background now. For the background, I want to select one lane. I'll open the lane profile tool, and we can see that this rolling disk size, the disk size of 10, is much too small for the background of this image. We can see it should start here, go flat across to here. So I'll again go up to the 70, which is again a very simple way to get a good background for very clean blots and gel images. And you can see that we get a nice flat background there. Let me click through and make sure that all of it works well for all the different lanes. And there's no problem there. So we've got the background set and we've got the lanes framed. And all I need to do is then Control C to copy the lanes and Control V to paste them on the other image here. And again, I'm going to need to click resize or adjust and just grab the frame and move it over so that it includes all the bands, gets it back in register. Okay, so now we've got the lanes framed and the background set. We go over to the bands. For this image, we're going to definitely use all of the bands in it, but for this image, we don't need to detect the bands at all. So on this image, I'm not going to detect any bands. I need total lane protein, and that's it. The exception to this rule is if there were a molecular weight standard in one of these lanes, then I would need to go over to the molecular weight standard tools and select that so that I could tell ImageLab which lane had the standard in it so that it's not included in the analysis. But there's no molecular weight standards here. So let's go back to this image, and I just need to detect the bands that I had before. So we'll go over to a detect band there and close that, and then I just need to delete all the bands that I don't want. So now I've got all my bands detected. I've got all the total protein that I need here, and we can go on to the final step, which is our normalization tool. We'll click over here, go to normalization. And in this case, the normalization channel is still our green channel, but it's the stain-free blot image. And this time we're using total lane protein as our normalization method. So now the data is ready. We'll open the analysis channel, our table. And because we have no bands detected here, the only bands that we see are the ones that we're interested in. Let me click on lane 1 here. So we see that the normalization factor was formed from lane 1, same as before. And so that normalized volume is going to be the same. Let me export this data again to Excel, and we'll make these bold and visible. There we go. So with total lane protein, what we did was we used the first lane of the total lane protein as our normalization factor. That created the normalization factor. Then every lane after that was used to normalize. So in this case, with total lane protein, you can see that the raw volume is being multiplied by a smaller number for the rest of the bands to get the normalized volume. What that means is that with total lane protein, the first lane actually has a lower protein load than all the other lanes. And this is the opposite trend that we saw with the actin. So with total lane protein, we get a different answer there. With that, I'll finish the demonstration of ImageLab normalization process. And we'll go back to our PowerPoint here. So the take-home message from this webinar is that there are many solutions to challenges in chemiluminescent blot analysis, and the TGX stain-free gels and ImageLab software provide tools for shortening protocols as well as strengthening data. So remember, when using a loading control, it's important to verify that the expression is consistent across all samples and treatments, and this is especially important when you're using a single housekeeping protein for a loading control. The stain-free gels provide a simple way to verify the blotting process also, as well as to normalize the chemiluminescent data. And ImageLab software provides tools to check the data and also to get the best analysis possible. Let's review the topics that we covered today. We've discussed many of the challenges with getting accurate data from Western blots. First, we looked at the 3 main challenges which fall into these broad categories: blot quality, image quality, and data quality. We then reviewed the solutions, including using TGX stain-free gels for verifying the blotting process, and ImageLab normalization tools to normalize the data. These tools can shorten the process and strengthen the data. We finished by going through the normalization process in ImageLab and compared using a housekeeping protein loading control with stain-free gels for a total protein loading control. I hope that you now have a good feel for working with ImageLab normalization tools to accurately analyze data from chemiluminescent blots. Thank you for listening. If you have any additional questions, please feel free to contact our technical support department at 800-424-6723 option 2. Either myself or one of my colleagues will be happy to further assist you.